Hi, this is my presentation for the Terminator region of AsdS sRNA in Streptococcus thermophilus. We chose Streptococcus thermophilus as our model organism due to its prominence as a lactic acid bacteria in fermentation products like yogurts and cheeses. Additionally, a very closely related organism is Streptococcus pyogenes, which is a pathogenic bacteria that causes flesh-eating disease. One of the reasons we used Thermophilus is that it's an excellent model for pyogenes' virulence, which is a measurement of how severe a disease can get or how far it can spread. On the diagrams below, you can see the secondary structure predictions, or how the organism's sRNA will fold, and you can see how similar they are both in some of the nucleotidal base pairings and the overall structure. An sRNA is a small regulatory RNA, and it's a non-coding RNA that regulates gene expression, either positively or negatively. One of the genes we're interested in looking at is the ASD gene in Streptococcus thermophilus, which serves as a biosynthesis pathway for multiple amino acids. One of the reasons we are interested in looking into the relationship between S. thermophilus and S. pyogenes is that in Thermophilus, directly preceding the ASD gene is the ASDS sRNA. In Streptococcus pyogenes, however, this gene is not present, but a highly similar directly preceding sRNA called MAR-S is very similar to ASDS. So we were interested in determining how MAR-S, which regulates virulence in Streptococcus pyogenes, could be so closely related to ASDS in the non-pathogenic S. thermophilus. In order to study the relationship between S. thermophilus and S. pyogenes in terms of virulence, we have to look at the transcription and termination processes more closely. DNA is transcribed into RNA, which is then translated into proteins. During the transcription process, an enzyme called RNA polymerase recognizes a promoter sequence and initiates the transcription process. After the transcription process is initiated, a transcription bubble forms. Within this, the DNA is pulled apart from its double helices and separated into two strands. RNA polymerase will then form an RNA strand to complement the coding DNA strand in a process known as elongation. During this process, the elongation is involved in synthesizing the RNA, and where the RNA and DNA meet is known as an RNA-DNA hybrid. Finally, in intrinsic termination, in Streptococcus thermophilus especially, the RNA polymerase recognizes a terminator structure, in this case a stem loop followed by a uracil string, and the RNase polymerase falls off and ends transcription. Taking a closer look into the transcription process specifically will also emphasize the importance of a transcription terminator. First, the intrinsic termination signal, which is the GC bonds and uracil string, is recognized. And the contrast between the three hydrogen bonds of the GC bearing and the comparatively weaker uracil bonds makes the RNA polymerase pause. This pause stops nucleotides from being added and gives the hairpin structure time to form. After the formation of a hairpin, it closes, and the place where the nascent RNA and DNA have been meeting, the RNA-DNA hybrid, melts. This causes structural instability and encourages the elongation process, which has been involved in the synthesis of RNA, to fall off and ends the termination process. As you can see from the termination process, this means that the transcription terminator is important in regulating the gene expression, as incorrect termination can lead to elongation complexes continuing to be read through the terminator. As we were working on the ASDS sRNA, we divided its structure into multiple different regions to be able to study them more closely. The first region, or the promoter region, contains the promoter that begins the transcription, and this part over here is called the cluster region. It contains a highly active and dissimilar part to the MAR-S sRNA in pyogenes, so we suspect that's where the virulence factors are located. However, this region or the Terminator region, is what controls the transcription termination of the AsdS sRNA. So the Terminator region is important due to its role in regulating how the termination of the sRNA is controlled. So it makes sure that it doesn't expend unnecessary energy on elongation, which will also regulate gene expression. So the AsdS sRNA terminator is an intrinsic terminator. That means it terminates independently of the Rho factor protein, as instead of having the protein regulate the structure of the stem loop, the RNA polymerase will recognize the structure of the stem loop and the trailing uracils after here, and instead it will decide to fall off and end termination properly. Another significant part of terminator structure is its conservation throughout Streptococcus species. The terminator sequence, compiled from 8 species, represents the structural and bonding overlays between species. Similarities in conservation in species is shown by the cleanly bonded pairs, like in the Terminator, highlighted by the blue brackets, with base pair types highlighted, as opposed to low conservation areas that often form unstructured loops and are represented with dashed lines. Species similarity is important due to how sRNA structure can determine characteristics like Terminator efficiency, whether the terminator is cis or transacting, and gene expression. The 3D prediction model of the terminator structure is good for visualizing the stem loop structure. Beginning with the blue pentose sugars on the left, the nucleotide sequences display the different base pairings in the sRNA. One significant part is the stem loop structure, with the guanine and cytosine pairings followed by the uracil string. Terminator strength, or terminator efficiency, is measured by the percentage of elongation complexes that are read through instead of cut off by an efficient terminator structure. Terminator efficiency is a key characteristic of the AsdS sRNA terminator due to its importance in preventing further elongation, which in turn regulates gene expression. Strong terminators have a GC loop and trailing uracil string and effectively terminate the sRNA, preventing it from expending unnecessary energy on elongation. While weak or inefficient terminators may also have a GC loop and uracil string, there are multiple complex causes of inefficient termination, such as upstream RNA structures, reaction conditions like concentration and variety of salts, whether the structure has been transcribed from supercoiling, and protein-dependent termination. For example, in E. coli, termination is dependent on the HFQ protein, which causes weak termination. In gram-positive bacteria like S. thermophilus, Nus-A and Nus-G proteins have the same effect, as termination reliant on these proteins is inefficient. In both strong and weak terminators, the efficiency can be positively or negatively regulated by any possible transacting sRNAs. As well as the terminator efficiency characteristics, whether an sRNA is cis or transacting impacts gene expression. When an sRNA is cis-acting, the genes and mRNA are on the same genetic locus, and there are regions of complete complementarity between the two. For transacting sRNA, a complementary mRNA bonds with the sRNA and impacts genes on different genetic loci. The complementary mRNA is less similar with the target gene than the cis-acting sRNA complementarity. The importance of transacting sRNA in termination is that a transacting mRNA may bond with the terminator structure and can either weaken the structure, causing inefficient termination, or strengthen the structure, causing more efficient termination. This is an example of how the RNA can regulate genes both positively and negatively. In conclusion, we can hypothesize that the AsdS sRNA is cis-acting on the ASD gene due to its closeness on the genetic locus, and it may be transacting on other genes or transacted upon by other RNAs. However, in order to determine this, we have to experimentally find out in the future. But in conclusion, the sRNA terminators are significant due to how they regulate the gene expression and the effective termination, And the cis or transacting traits are significant in that they regulate which genes are being impacted and how that also affects termination as well. In order to experimentally prove terminator strength, you can test the terminator efficiency through terminator read-throughs. And what these do is that they measure the elongation complexes that are present in the transcription process, and they measure how many of them and what percentage pass through the terminator efficiency using the fluorescent intensities of the elongation complexes that have been marked. In order to experimentally determine whether the sRNA is cis or transacting, you would use fluorescence in situ hybridization to determine the RNA's impact on the mRNA sequence. So how this works is that the mRNAs are radioactively marked, and then afterwards, once they've bonded with their target RNAs or genes, then they'll show up in fluorescence, and that'll demonstrate the gene expression as well as the mRNA location. So you can see that'd be a valuable tool for determining whether an RNA is cis or transacting and what it's acting upon. Thanks to all the people and programs who made the summer research possible, and thank you for watching.